We are entering an AI drug revolution, and today, targeted protein degradation, specifically PROTACs and molecular glues, is finally explained. For decades, traditional pharmacology was stuck in the lock and key model, where drugs simply had to continuously block a bad protein's active site. But what if we could build event-driven molecular assassins that tag these disease-causing proteins and drag them straight to the cell's natural garbage disposal? By hijacking the Ubiquitin-Proteasome system, we can now target the 85% of proteins once thought completely undruggable. Today, we are unlocking the exact mechanisms of how PROTACs and molecular glues are rewriting the rules of medicine. Let's look at the blueprint of this paradigm shift. For decades, traditional drug design relied on an occupancy-driven model. This meant a drug had to continuously occupy a target's active site to inhibit its function. It's a constant physical blockade. If the drug lets go, the blockade fails and the target goes right back to work. But we're moving beyond this traditional design. Emerging technologies use an event-driven mechanism. A single drug molecule triggers a permanent cellular event, like total destruction, and then moves on to catalyze the exact same reaction again. To understand how we drug the undruggable, we have to look at the old physics of pharmacology. Traditionally, drugs relied on an occupancy-driven model. This is strictly stoichiometric. One drug binds to exactly one target. Because it requires continuous binding in a deep pocket to work, the drug gets completely used up on a single target. It's highly inefficient. But the new physics of event-driven pharmacology changes the math. Instead of continuous binding, it uses a catalytic hit-and-run mechanism. One drug molecule can tag and destroy multiple targets. This is how we finally access the 85% of the proteome we used to call completely undruggable. Before we get to total protein destruction, let's look at how we are refining traditional targets. Historically, activating a receptor was an all-or-nothing event. As you can see here, a traditional agonist acts like a scattered prism. It triggers the therapeutic pathway, but it also triggers the side effect pathway at the same time. You couldn't get the efficacy without the on-target toxicity. To get pain relief, you had to accept the respiratory depression. This is where biased agonism, or functional selectivity, changes the game. By stabilizing specific receptor conformations, these new drugs act like a focused laser instead of a scattered prism. They selectively activate one pathway while completely silencing the other. For example, the opioid oliceridine is designed to selectively activate the G protein pathway for pain relief while actively avoiding the beta-arrestin recruitment that causes tolerance and respiratory depression. But what if we don't want to turn a receptor fully on or off? This brings us to biased allosteric modulators, or BAMs. Traditional drugs bind directly to the primary or orthosteric active site. The problem is that these active sites look almost exactly the same across different protein families, which creates a huge risk for off-target side effects. Traditional blockers act like a blunt off switch. They completely shut down the signaling pathway, disrupting the body's natural physiological rhythm. BAMs take a smarter approach. They bind outside that main pocket, targeting unique, highly selective allosteric sites. Because these sites are less conserved, BAMs can achieve incredible subtype selectivity. Instead of an on-off switch, BAMs act like a precision volume knob or a dimmer switch. They fine-tune the signal while perfectly preserving the natural rhythm of your endogenous hormones. Now we reach the ultimate event-driven therapy: Targeted Protein Degradation, or TPD. Instead of just temporarily blocking a protein's function, TPD physically eliminates it entirely. It does this by hijacking the body's natural waste disposal machinery, the Ubiquitin-Proteasome system. Why is this necessary? Because many disease-causing proteins, like structural scaffolding or toxic aggregates in Alzheimer's, don't have active sites. Traditional drugs literally have nowhere to bind, so they just bounce right off. TPD bypasses this by acting as a protein demolition crew. The drug forces the bad protein next to an E3 ligase, which slaps a chain of ubiquitin tags onto the target. In molecular biology, this is literally known as the kiss of death. The cell's own shredder, the proteasome, recognizes the tag and pulverizes the protein into harmless peptides. And the best part? Step 4, recycling. After the target is destroyed, the drug molecule survives perfectly intact, Moving on to clear out the next toxic protein. So, how do we actually force a target into the cell's disposal system? Enter PROTACs, or proteolysis targeting chimeras. These are massive heterobifunctional molecules weighing over 800 Daltons. They act like a double-sided molecular handcuff. A warhead binds the target protein, an anchor binds the E3 ligase, and a linker ties them together. This forces the two proteins into a ternary complex, triggering proximity-induced ubiquitination. But their massive size causes complex pharmacogenetics. They are also vulnerable to the hook effect. If the drug concentration is too high, the PROTACs just competitively bind to everything, which actually reduces their efficacy. Despite their bulky size, their therapeutic edge is undeniable. Because they are highly catalytic, they can tear down mutant proteins that completely resist standard inhibitors. We are already seeing this clinical validation with drugs like vepdegestrant, which actively degrades mutant estrogen receptors in breast cancer. The cancer mutates to block the drug, but the PROTAC just grabs it anyway, shreds it, and goes right back for the next one. While PROTACs are incredibly powerful, their huge size makes it difficult for them to reach certain areas, like the brain. This brings us to our next modality, molecular glues. Unlike PROTACs, these are small monovalent molecules weighing under 500 Daltons, and they have absolutely no linker. Normally, an E3 ligase might completely ignore a disease protein because their molecular shapes simply do not fit together. To the cell's garbage disposal system, that target is effectively invisible. Molecular glues solve this by binding directly to the E3 ligase and physically reshaping it, creating an induced neosurface. This allows the ligase to suddenly grab and recruit novel targets it would never normally bind to. And because these glues are so small, they offer superior oral bioavailability and excellent blood-brain barrier penetration. A classic example is thalidomide and its analogs, which serendipitously act as glues to drag cancer-driving transcription factors straight to the shredder. Historically, finding these powerful molecular glues relied almost entirely on serendipity, or pure luck. But the computational revolution has completely flipped this process. Trying to manually find a drug that perfectly forces a disease protein and an E3 ligase together is like trying to solve a billion-piece puzzle in the dark. The traditional drug discovery timeline takes years of trial and error. Now, artificial intelligence is doing the heavy lifting. AI tools like AlphaFold predict the 3D structures of undruggable targets, while generative platforms like PandaOmics and Chemistry 42 map the exact interactions needed to create a neosurface. In pilot programs, this AI toolkit has reduced end-to-end drug discovery from years down to just 30 days. AI can identify ligand binding sites that only exist when two proteins are brought together, turning what used to be a lucky accident into precise, calculated engineering. So we have these incredible protein-shredding PROTACs, but there is a major catch. Because they are so massive, they often have terrible solubility and struggle to distribute properly through the body to reach the tumor. If this potent molecular assassin gets lost in the bloodstream or drops its payload in the wrong tissue, it can cause severe systemic toxicity. It needs a ride, and it needs a VIP pass to the tumor. Enter advanced conjugates, specifically degrader antibody conjugates, or DACs. We use a highly specific antibody as the guide, or the bus, and we attach the PROTAC as the passenger using a cleavable linker. The antibody navigates directly to the tumor cells, bypassing healthy tissue entirely. Once inside the tumor, the linker is cut, releasing the PROTAC to unleash its catalytic protein-destroying power right where it's needed. This solves the delivery issue and completely overcomes traditional drug resistance. Let's move from molecules to living cells. Traditional CAR T-cell therapy is a logistical nightmare. It requires extracting a patient's blood, transporting it to a lab to manufacture the cells over several weeks, and then reinfusing them. It is incredibly slow and costly. Because of this massive vein-to-vein time and the absolute reliance on specialized manufacturing facilities, access to this life-saving therapy is severely bottlenecked. The solution is in vivo CAR-T. Instead of removing the cells, we use targeted vectors to deliver the genetic instructions directly to the T cells inside the patient. The patient's own body literally acts as the bioreactor, reprogramming the immune cells on the spot. This completely democratizes the therapy, and it's allowing us to expand beyond just blood cancers to actively treat autoimmune diseases like lupus by resetting the entire immune system. Finally, we are entering the era of truly programmable modalities. We can now give the body extreme binary instructions, either build or destroy. In the past, treating something like cancer with radiation or systemic drugs was like carpet bombing a city. The collateral damage to healthy cells was devastating. Now we have two surgical options. First, the code. We use mRNA not just for infectious diseases, but for personalized cancer vaccines. We encode patient-specific tumor neoantigens to prime the immune system to hunt down the cancer. Second, the physics. Targeted radioligands. We take a radioactive isotope and attach it to a molecule that only binds to specific cancer markers like PSMA. It delivers highly localized, cell-destroying radiation directly to the target, completely sparing all the healthy tissue around it. With all these massive advancements in PROTACs and cellular therapies, our old testing methods are completely breaking down. Traditional 2D cell cultures cannot predict the complex immune responses or tissue penetration required for these next-gen drugs. Complex mechanisms like biased agonism and TPD simply fail in 2D models. If you test a 3D robotic assassin in a flat 2D environment, you get useless data. The solution is to test in a human-relevant physiological context before we ever reach clinical trials. We are now using 3D patient-derived tumor organoids that retain original tumor heterogeneity and organ-on-a-chip technology that accurately simulates blood flow and organ interactions. These microfluidic chips act as living simulations, ensuring that when these drugs finally enter a human, they do exactly what they were engineered to do. We have officially entered the era of pharmacological orchestration. From simple occupancy-driven inhibition to AI-designed molecular glues to permanently deleting disease proteins, we are no longer limited by the druggable genome. But now that you understand how these revolutionary drugs are engineered to destroy disease, you aren't ready for the future of medicine until you see how your own DNA decides if these drugs will actually work on you. In this video right here, we are breaking down pharmacogenetics, exploring how specific ADRB2 gene variants totally change asthma treatment, and how we map your DNA for true individualized therapy. Click here and let's unlock the final chapter of your pharmacology lore.